Okay. So for 2.7, I believe it is, which is active transport, we're going to talk about how things move from an area of low concentration to an area of high concentration. Think about moving a boulder uphill or a bowling ball or something uphill or even just walking uphill. Going from an area of low concentration to high concentration against the concentration gradient is going to require energy. And the energy can come in various forms. One of those forms is ATP, and that's something like a sodium potassium pump is an example of one of those ATP dependent mechanisms. But then we'll also learn about some processes that we use just the general energy pool of the cell. Okay. So usually with some molecules, it's usually that they're going to be too charged to get across. Um, they're going to be moving from an area of low to high. So that's going to be the need for something like a sodium potassium pump here. Sodium potassium pump is very important for your nerve cells, your brain, your nerves. Very, very important for that. I mean, it's important in a lot of different cell types, but I would say nervous tissue, brain cells, muscle cells, those are going to be more reliant on that. Think of cells that are using a lot of energy. Okay, so let's look at how the sodium potassium pump. This is going to be like a cartoon version of it. Um, it is based on shape and in charge on the protein, but again, it's not going to be like 3 circles to 2 triangles. But they're just showing you that for sake of understanding how that's happening. So make sure you understand what's happening with sodium, what's happening with potassium, what's happening with ATP, what's required, this membrane protein, and very important to pay attention to the gradient. Is it going from low to high or high to low? That's very important.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. One phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
Okay, so just to reiterate that, 3 sodium out for every 2 potassium in, but first the expense of 1 molecule of ATP. All right, one thing that this is used for is to generate what are called membrane potentials across biological membranes. And specifically, we're going to talk about the cells that are most reliant on this, or, it's probably that it's just been most studied, that we understand more about that, but that would be in nerve cells. And then, so let's look at this animation.
I've said before before that a nerve impulse is an electrical signal that travels along the neuron, but it's not quite electricity as you may know it. Electricity is the flow of electrons, but in a nerve impulse we use the word potential to describe the movement of charged particles across the membrane of a cell. The charged particles flow like a wave down the axon, moving in and out of the cell. The movement is mostly of positively charged ions, not negatively charged electrons like you might imagine. Let's look even closer at the cell membrane. At rest, when a neuron isn't firing, there's a high concentration of sodium ions outside of the cell membrane, and also a high concentration of potassium ions in the cytosol of the cell. The sodium and potassium channels are gated and closed. Because of the difference in number of sodium and potassium ions, as well as the presence of negative ions like chlorine, there's an overall negative internal charge of negative 70 millivolts. The beginning of an impulse is marked by depolarization. This means that the voltage-gated sodium channel is opened and the sodium rushes into the cell. The influx of positive charges quickly shifts the electric potential of the membrane so that now the outside is more negative and the inside of the cell is more positive. This change in polarity will close the sodium gate. Nearby is the voltage-gated potassium channel, which will now open and allow potassium ions to flow out of the cell. The movement of these positive ions will repolarize the cell, allowing the cell to have a negative internal charge relative to the positive extracellular charge. The change in polarity will also close the potassium gate. At this time, you might think that the cell is ready to receive another signal, but it can't. If the sodium gates opened, the sodium ions would not move. In order for ions to move through their channels, they have to be able to move down the electrochemical gradient. The positive sodium ions aren't attracted to the positive environment outside of the cell, so they won't move. Another signal can't be sent until the sodium and potassium ions have moved back to their resting potential positions. But this means moving against concentration gradients, which means energy must be used. During the refractory period, the sodium-potassium pump will move 3 sodium ions out of the cell and 2 potassium ions into the cell with 1 ATP molecule. This must happen constantly and rapidly, so the brain uses a significant amount of your body's energy. In a matter of milliseconds, the sodium-potassium pumps will restore resting potential and another action potential can move through. Nerve impulses are actually waves of action potentials, which means ions are flowing in and out of the cell in a wave-like pattern. Because of the refractory period where sodium ions will not flow out of the cell by diffusion, action potentials can only move in one direction. They begin at the dendrites and they end at the axon terminals. This wave-like region where the action potentials are flowing are referred to as the local current. The flow of sodium through one gate will trigger the flow of sodium in the next gate, but sometimes there isn't enough sodium to trigger an action potential.
Hence is the reason why, if you don't have enough sodium, this all stops. Your brain stops. So that's one reason why lack of sodium is crucial.
Using an oscilloscope to display the membrane potential over time, we can see that sometimes the flow of sodium ions isn't enough for an action potential to occur. The internal region of the cell must reach negative 55 millivolts, the threshold potential, for action potential.
We don't need to know the specifics of action potentials, at least at this time. I don't think it's in the curriculum for AP Bio.
If enough sodium ions are released to surpass the threshold potential, an action potential occurs. In the oscilloscope trace of an action potential, we can see where the sodium and potassium gates have opened. Here is the point where the stimulus occurs, and sodium gates open, allowing sodium to rush into the cell. This depolarizes the cell, causing the inside to become positive relative to the outside of the cell. This polarity change opens the potassium gate, which allows potassium ions to rush out of the cell, which rapidly repolarizes the inside to a negative charge. This can often become more negative than negative 70 millivolts and is called hyperpolarization or undershoot. This will be corrected during the refractory period, where the sodium potassium pump uses ATP to restore resting potential.
Okay, I think you want to be able to differentiate. For our purposes, I want us to know sodium potassium pump. That's to get it back to the point. So in that case, it's going to be 3 sodium out, 2 potassium in, which is going to get it set up for the next run.
During this time, no additional action potentials can be sent. But once resting potential is reached, the neuron is ready to fire again. Thanks for watching this episode of Teacher's Pet. Don't forget to like and subscribe and follow me on Twitter at SciencePet.
Okay. So that just kind of showed a everyday example of how sodium potassium pump is important. It restores the gradient. It has to go against the concentration gradient, so it's going to require energy. 3 sodium out, 2 potassium in at the expense of 1 molecule ATP to set that back up. Okay, so we talked about these are active transport mechanisms. This is just one example. There's lots of others. This is probably the most well described and understood. So in this case, ATP is required directly. Like if you don't have ATP, you try to it's like they say cash only and you try to pay with a card. Well, they don't have a card reader. It's not going to work. So same thing with sodium potassium pump. If you try to use NADH or something, it's not going to work. It needs to have ATP. Okay. This next type of transport mechanism, and again, I think this is mostly review, so I'm just going to quickly go through it, is going to be called secondary active transport because, yes, energy is required, but no, it doesn't have to be in the form of ATP. It can be, but there's other energy currencies of the cell. Just like, I might use a cash, check, credit card, debit card. Same thing in the cell. They have ATP, NAD, NADH, other examples as well, FAD. And that energy could be used in these processes as well. So I guess the only thing I want to say about endocytosis and exocytosis is, I guess, just the basics. Just for review. So phagocytosis is going to bring in solid particles. So it could be things like cells or just small amounts of cellular debris. Sorry about that. Scrolling. Okay. And then pinocytosis is for fluids. So all 3 of these are endocytosis, and then I don't have exocytosis here, but it's basically the reverse. And I'm going to probably bring up a couple animations. Pinocytosis is to bring in pockets of fluid. So let's say that there's fluid that has a lot of dissolved sugars and things. It can bring in kind of take sips out of it. So phagocytosis is like eating. Phage means to eat. Pino is more equivalent to drinking, taking little pockets of fluid. And then receptor-mediated endocytosis is a specific type of endocytosis where the membrane is going to be coated with receptors, and then they're going to form a pit and bring in a specific substance. And the example that I always require for receptor-mediated endocytosis is cholesterol. That's how cholesterol is going to come into the cell. And let me see if I can find the animation for that.
Endocytosis is a type of vesicular transport involving the active movement of substances into a cell in membrane-bound vesicles. There are 3 main types of endocytosis: receptor-mediated endocytosis, phagocytosis, and pinocytosis. Receptor-mediated endocytosis occurs when a ligand binds to a specific receptor on the cell's plasma membrane, forming a ligand-receptor complex. Each receptor is associated with a protein known as a clathrin on the membrane's cytoplasmic side. The regions of the plasma membrane in which these receptors and their associated clathrin molecules are expressed are known as clathrin-coated pits. The clathrin-coated pit sinks into the cell, forming a vesicle that contains the ligand-receptor complexes. Once in the cytoplasm, the clathrin molecules coating the outer edge of the vesicle leave and associate with new receptors on the plasma membrane. The uncoated vesicle fuses with an endosome, and the ligands and receptors separate, collecting at opposite ends of the endosome. Sections of the endosome containing unbound receptors pinch off, forming transport vesicles that return the receptors to the plasma membrane. The remaining vesicles, which now contain free ligands, fuse with a lysosome containing digestive enzymes. Finally, the lysosome's digestive enzymes break the ligand down into smaller molecules, which are then released into the cytoplasm of the cell for use in a number of cell processes.